Welcome everyone. In this video, we'll discuss few practice problems related to genomics, genetics, and these questions are quite common in several of the competitive exams like CSIR NET, DBT-JRF, ICMR, and GATE. Let's discuss the first question. It has been asked to calculate C value in picograms for Zia maize, and the total number of DNA present in a diploid cell is 4.6 billion base pair. And we all know the C value. How do we define the C value? It's the DNA content present in a haploid cell, but the information which is given is 2n. So the n is going to be 4.6 into 10 raised to power 9 divided by 2, which in this case is going to be 2.3 into 10 raised to power 9 base pair. But we need to calculate the C value in picograms. So we need to make some conversions. We all know the average molecular weight of a base pair is, in case of DNA, is 660 gram per mole. And we have total 2.3 into 10 raised to power 9 base pair. So in this case, this is going to be 2.3 into 10 raised to power 9 base pair into 660 gram per mole. And remember, 1 mole is, we all know it's a Avogadro's constant. So that is going to be, in this case, 6.023 into 10 raised to power 23. So these are the numbers. You can make use of the calculator to calculate it. And if you'll try to solve it, you will get, I'm making use of my calculator. So the numerator will become 1518 into 10 raised to power 9. And then in the denominator, you have 6.023 into 10 raised to power 23. And the unit will be gram. And this will come 252 into 10 raised to power minus 40 gram. Now, let's get back to the question again. It has been asked to calculate the C value in picogram, and we all know 1 gram equal to 10 raised to power 12 picogram. So this much gram, how many picograms? 252 into 10 raised to power minus 14 into 10 raised to power 12 picogram, which corresponds to 252 into 10 raised to power minus 2 picogram, which is nothing but the 2.52 picogram. So this is going to be answer. You can either report 252 into 10 raised to power minus 2 picogram, or in a better representation manner, you can call it as a 2.52 picogram of, this is the C value, which is going to be for Zia maize. The only thing which you need to be very careful is we define C value as a haploid DNA content, and that's the reason I made this conversion at an initial point. And then you need to consider the average molecular weight of a base pair, 660 gram per mole. And the other thing which you need to consider is 1 mole is 6.023 into 10 raised to power 23. It's a Avogadro's constant, so it comes in 1 mole of a DNA, those many molecules of DNA. Let's move to the second question. And this is a case of a paternity test or a question from the DNA forensics. Two men claimed to be the father of baby Caroline. Caroline's mother had her CODIS STR, which is again a locus combined DNA index system. Short tandem repeats DNA profile analyzed and was homozygous for allele A. That means both the two copies are the same at this particular locus, TPOX locus, and allele 8 contains 8 repeats of GAAT sequence at this polymorphic locus. Information given. Baby Caroline is heterozygous for alleles 8 and 11 at this locus. So mother is homozygous for allele 8, and baby Caroline is heterozygous for alleles 8 and 11. So if you can read this question carefully, it should be very clear that Baby Caroline is getting allele 8 from her mother. Clear. In an attempt to resolve the disputed paternity, the two women were tested for their STR DNA profiles at this particular locus on chromosome 2. Putative father 1 was heterozygous for alleles 8 and 11 at the TPOX locus, and putative father was homozygous. Putative father 2 was homozygous for allele 11 at this locus, and you need to solve the case of disputed paternity and who is the biological father. You need to explain. So let's try to understand this question. Let me draw a pattern and then we can discuss. So remember, Caroline mother, baby Caroline, and then you have the putative father 1 and putative father 2. As we read, mother is homozygous for both the allele, and what you will get in an ideal situation when you are doing marker tests like molecular marker tests like RFLP or AFLP, you'll get a single band. The reason being both the two copies are in a homozygous state, or rather, Caroline's mother has, she has got both the copies of the allele from her parents. So clear. Now, baby Caroline is heterozygous, and she is carrying one 8 and the other is allele. And it should be very clear that this 8 is coming from her mother. So this one is 8. So you can consider this band corresponding to allele 8, and the other allele is allele 11 in case of Caroline because she is heterozygous. Now putative father P1, even P1 is heterozygous. So as you can see, putative father 1 was heterozygous for alleles 8 and 11. And we all know that the 8 in Caroline is coming from her mother, and the putative father 1 is again a heterozygous for allele 8 and 11. So this, one can think of that, not conclusive. He might give his allele 11 to Caroline. Let's look for the other case. Father 2, putative father 2 was homozygous for allele 11. So in that case, you will get band, a single band, because again, he is homozygous. And even in this case, there is a possibility that he might give allele 11 to baby Caroline. So we cannot say conclusively who is the possible father. It could be P1, P2. The data are insufficient because remember, baby Caroline is getting 8 from her mother, 11. She could get it from either from P1 or even from P2. So coming back to the question, can these results resolve this case of disputed paternity? No. And who is the biological father? We cannot conclude from this data. The data is insufficient. And as explained, baby Caroline has to get 11 and she could get it either from P1 or P2. Remember, offsprings inherit one chromosome from the father and the other from the mother. Now, what is the strategy to conclude who is the biological father? You need to look for other CODIS locus. Remember, there are almost 12 CODIS locus, and what you can do, you can target other locus and can confirm who is the biological father. We all know what are these locus. These are the repetitive DNA sequences, and which is usually unique in the individuals. So CODIS stands for Combined DNA Index System. So what we generally do, we target few of the unique locus which are present in an individual, and then we try to figure it out who is, then we try to solve the cases corresponding to the DNA forensics, be it any criminal cases or paternity or any other cases because we all know that an offspring inherits one chromosome from the father and the other from the mother. Let's move to the next question. If a human nucleus is 10 micrometer in diameter given, and it must hold as much as 2 meter of DNA, which is complexed into nucleosomes that during full extension are 11 nanometer in diameter, what percentage of the volume of the nucleus is occupied by the genetic material? We all know inside the cell, we have the nucleus, and within the nucleus, we have 46 pair of chromosome, 22 autosomes, and one pair of sex chromosome XX in females, XY in males. Now read the question carefully. If a human nucleus is 10 micrometer in diameter and it must hold as much as 2 meter of DNA, which is complexed into nucleosomes that during full extension are 11 nanometer in diameter. We all know, the organization of a eukaryotic genome. We have, the 2 nanometer diameter DNA, which is complex with the histone proteins to form nucleosomes 11 nanometer structure, which further complex to form 30 nanometer chromatin fiber, further condensed and complex to form 300 nanometer chromatin fiber. And then you have a chromatids of the chromosome, which is close to 700 nanometer. And both the two chromosomes are, both the two chromatids are 1400 nanometer structure. What you need to calculate is what percentage of the volume of the nucleus is occupied by the genetic material. So volume of nucleus, first you need to calculate, which is nothing but a volume of the sphere. And in this case, you can calculate this via the formula 4 by 3 pi r cube, correct? Which in this case is going to be 4 by 3 into 3.14 into r. R given is diameter given is 10 micrometers, so R is going to be 5. And remember, since the DNA, the size of the nucleosome is in the nanometer, we should convert it into a nanometer. So 5 micrometer corresponds to 5,000 nanometer and then cube. I hope this is clear to each and every one of you. And if you'll try to calculate, you will get it. You can make use of the calculator to calculate it. 5.23 into 10 to the power 11 nanometer cube. So this is the volume of the nucleus. Now the second thing which we need to calculate is the volume of this particular complex, which is a structure of nucleosome. We all are aware what is nucleosome. Nucleosome is an 11 nanometer structure consisting of DNA wrapped around a histone protein. The histone is made up of two copies of H2A, the core nucleosome I'm talking about, two copies of H2A, H2B, H3, and H4, and H1 connects one nucleosome to the another. H1 is not a part of a core. So you have a DNA wrapped around this core nucleosome, which I'm calling it as a histone protein. Now, we need to calculate the volume occupied by this structure. So we all know the if you look at the chromosome, if you try to pull it, you'll get a 2 nanometer structure, which is complex to form a 11 nanometer structure. And the volume of this material is, you can calculate that one by volume of nucleosome is looks more like of a cylindrical structure. So that is going to be pi r square h, and in this case, it's going to be 3.14 into 11 nanometer is a diameter. So that is going to be 5.5 square. H is this 2 meter of the DNA. So you have 2 meter of a DNA, which is complex into nucleosomes. So if you look at this entire structure, remember we have, you know, 30 nanometer structure corresponds to, the two nucleosomes will be joined together adjacently. I mean, more and more nucleosomes will be joined together with the help of this linker DNA. As said, the linker DNA connects the two nucleosomes, and the H1 is present close to this end. It is not a part of a core nucleosome, but it helps in connecting the adjacent nucleosomes. So H given is 2 meter of a DNA. Remember, it's a 2 meter, so we should convert it into a nanometer. And if we'll make use of the calculator to calculate this value, we'll get the number which you should get is 1.89 into 10 raised to power 11 nanometer cube. We have calculated the volume of the nucleus considering it as a sphere, and then we have calculated this one as a volume of cylinder. The chromosome, we all know the structure of the chromosome. So this is the 1400 nanometer structure, and at some point, if we'll try to open it, you will get a 300 nanometer structure. This is the same thing I'm talking about. Which you further pull it, you will get a 30 nanometer, and then we all know if you keep on pulling it 11 nanometer and until you get a 2 nanometer diameter DNA, correct? Now the question which has been asked is what percentage of the volume of the nucleus is occupied by the genetic material? So what we need to do is what percentage of the volume of the nucleus is occupied by the genetic material, which is the DNA, and that has to be 1.89 into 10 raised to 11 nanometer cube divided by 5.23 into 10 raised to 11 nanometer cube. Since the percentage is asked, we need to multiply it by 100 and we get close to 36.13%. So the answer is going to be 36.13%. So this percentage of the volume of the nucleus, 36.13% of the volume of the nucleus is occupied by the genetic material, which is the DNA. And we all know that is complex in the form of a cylindrical structure, which is a chromosome. Let's move to the next question, question number 4. A previously undiscovered single-cell organism was found living at a great depth on the ocean floor. Its nucleus contains only a single linear chromosome consisting of 7 into 10 to the power 6 nucleotide pairs of DNA, coalesced with 3 types of histone-like proteins. Consider the following questions. So there are 2 parts. The next question is in the next slide. A short micrococcal nucleus digestion yielded DNA fractions consisting of 700, 1400, and 2100 base pairs. Predict what these fractions represent and what conclusions can be drawn. We all know the famous experiment of micrococcal nuclease digestion. It's a nuclease which digests the phosphodiester bonds present between the adjacent nucleotide. Remember, the treatment given is for a short duration. So what will happen? We all again get back to the same structure, nucleosome. And nucleosome, as said, is a complex of a DNA and protein. And depending upon what is the length of the linker DNA, is you'll be nucleosomes lying together to form these 30 and 30 nanometer chromatin fiber like structure. So you'll see something. Now remember, when you are giving a micrococcal nuclease treatment, what it does, it cleaves the phosphodiester bond present in the DNA. And if you can see, the only DNA which will be cleaved is the DNA which is connecting the two nucleosomes. The DNA which is wrapped around, will not be cleaved. And the reason is simple, because that DNA is protected by the DNA is protected by the histone protein. So that is a part of a core nucleosome. and microfocal nucleus will not be able to access. That DNA is not naked, that DNA is in a complex form. So this will not be digested. Whatever will be digested is the linker DNA. And remember, the length of the linker DNA varies. So depending upon how, whether you are dealing with a euchromatin or a heterochromatin. So where the DNA expression is, sorry, where there is a more gene expression, you will find the DNA is more loosened up and will get expressed more compared to where your gene is not getting expressed, which is not given there in the question. What we can consider is, the length of the linker DNA is not significant enough. Now, getting back to the question, a short micrococal nucleus, remember it's a short. So what will happen is, there will be cuts taking place, and I mean, there is a possibility that there is a one cut here. In the another ideal situation, there is the first cut is here. In the third situation, there is a cut here. And what you can figure it out by looking at these DNA fragments, that these are in the multiples of 700, 700, 1400, and 2100 base pairs. So the first thing which should come into your mind is that the DNA, which is, I'm calling it as a core DNA, which is wrapped around a histone-like protein, is close to 700 base pair. Correct? Because these are the ones which will not be digested at all. Even if we give short or extended period of a digestion, this is not going to be cleaved, because these are the DNA which are protected. Now, as said, so you will get, even at extended period of a digestion, 700 base pair of a DNA. And depending upon the cut site, depending upon the probability where there is a cut taking place, and since the duration of the cut is short, you will get a situation where there is a cut here which is close to 700. In the second case, you will get a cut somewhat not here, but directly here. So 700 plus 700, 1400. In the third case, you are getting a cut only at this place, which is 700, 1400, and 2100, so on and so forth. So predict what these fractions represent. You can write that the DNA, which is wrapped around nucleosome, the DNA, which is a part of a core nucleosome, is 700 base pair, and these numbers are in the multiples of 700, suggesting that the digestion is not complete. And these are the fragments of a DNA which you'll find on a first first nucleosome, second nucleosome, and the third nucleosome, so on and so forth. What conclusions can be drawn? One of the major conclusions which you can cite here is the wrapped DNA length is 700 base pair. I mean, each nucleosome in this particular case is wrapped or is wrapped or is consisting of 700 base pair of DNA, unlike humans where we see 160 base pair of a DNA wrapped around a histone protein. Correct? Second question, second part of the same question. The analysis of individual nucleosomes revealed that each unit contained one copy of each protein. So analysis of individual nucleosome revealed that each nucleosome unit, contained one copy of each protein. And we all know in the first part of the question, it was given that DNA coalesces to it three types of histone-like proteins. So each nucleosome is consisting of three types of protein. XL1, we can call it as histone-like 1 protein, histone-like 2, histone-like 3. So the analysis of individual nucleosomes revealed that each unit contained one copy of each protein. So one copy of XL1, XL2, XL3, and that the short linker DNA had no protein bound to it. I mean, even here they have given the short linker DNA. If the entire chromosome, linker I'm talking about, the DNA which connects the two nucleosomes. This is the wrapped one is 700 base pair. This is going to be the same for all the nucleosomes. If the entire chromosome consists of this nucleosome-like structure, which is a complex of DNA and histone protein, and they are discounting any linker DNA, how many are there and how many total proteins are needed to form them. Interesting. So the length of the DNA which is given is, sorry, 7 into 10 raised power 6, correct? Base pair. And the DNA which is wrapped around this structure is 700 base pair in length. So how many nucleosomes will be present? That is going to be 10 raised power 4 nucleosome. I hope this is clear to each and everyone. The entire chromosome given, I mean, they have mentioned consists of nucleosomes. How many are there? 10 raised power 4 nucleosomes, and how many total proteins are needed to form? We all know the one, the single structure, as you can see, is consisting of this. This particular nucleosome structure is consisting of 3 histone proteins, HL1, HL2, HL3. So 10 raised power 4 nucleosome will consist of how many? Simple. 10 raised power 4 into 3, which is 3 into 10 raised power 4 proteins. Nucleosomes. I hope the each and every question is clear to you. If not, please do not forget to comment in the comment section if you have any kind of issues. And also, if you do have any kind of such questions, you just share with me. I'll be more than happy to take it. Take care. Thank you very much.